Thank you so much for having me. As a scientist, I don't get too many opportunities to interact with the families and the patients, so it's a real treat for me when I get to come to the conferences. I have to move this down a little house taller than me. Okay, so today I'm going to tell you about some research that we've been doing. This is really a summary of work we've done, I would say, probably over the past 5 years or so. Let me see if I can get this technology to work. Apparently not so much. Okay, so this is kind of the outline of my talk. I'm going to quickly move through background, some of which you may already be comfortable with, and spend the bulk of the time talking about what we've actually done and what we found, and then just follow up with some concluding remarks. So, why do we think it's possible that there's a genetic basis to Chiari malformations? Well, the simplest thing is just to look within families. So we do have examples where there are families who have multiple individuals who are affected with the condition. And I know some of you are sitting out there right now. We also have information from twin studies and even triplet studies where what we have been able to observe is that there's a correlation, a higher concordance of Chiari malformations in identical twins or identical triplets. And there's also this thing in genetics like guilt by association, so to speak. And that's where we see that Chiari malformations often co-occurs with other clinical conditions, which we already know has a genetic basis. And then from the Duke family study many years ago, we had already proven that there were regions on chromosomes 9 and chromosomes 15 that may harbor genes for Chiari. And moreover, what I'm going to spend a lot of time talking about today is that this was the first study to show that the morphology of the back of the skull, the posterior fossa region, is actually heritable, regardless of whether you have Chiari malformations. So, this is great. We know that there's probably a genetic basis, at least in a portion of the families, but there are a lot of hurdles that we have to overcome as genetic researchers. So the first is, and we'd already talked about this a little bit in some of the morning sessions, is that Chiari malformations tends to be a little more rare than a number of conditions. And so if I want to study the genetic basis of Chiari malformations, it can be quite difficult to collect enough individuals just by nature of how frequent the condition is. And then, moreover, if we look at all the Chiari patients, probably only about 10 to 15% of those individuals have another family member who also has the condition. So it's actually, a rare condition, and then even a smaller proportion of those individuals have a heritable form, at least as best we can tell. So that's some of the hurdles we have with just kind of collecting family members. But there's also challenges with respect to the diagnosis, or as a geneticist, I'll call it the phenotype, because I'm not a clinician. But I'm sure you've heard a lot of discussion throughout yesterday and today. There's no consensus diagnostic criteria, which makes it difficult because if you're trying to compare results from one study to another, sometimes people use different criteria to define who has the condition and who doesn't, and that makes it very difficult because you're comparing apples to oranges. One of the other things is that we know tonsillar herniation does not always correlate well with symptom presentation, but we use tonsillar herniation most of the time to define who's affected and who isn't. And one of the ways we really, especially as geneticists, like to get a much more objective picture of what's happening in individuals is to look at the MRIs. And as I mentioned earlier, just sort of look at the morphology in the back of the skull. But it can be very difficult to obtain a lot of MRIs. And then at the end of the day, what we think is we really have probably a group of conditions in Chiari malformations. There's probably multiple genetic factors, multiple environmental factors, and so basically if I've collected a set of families to examine and find a genetic basis, I'm really focused on this fruit basket, if you will. There's oranges and apples and grapes, and it can be very difficult to pull out a subset of families and individuals who may be sharing the same gene or same etiologic cause. But undaunted, we forged ahead. And so one of the first things that we did was we conducted what's called a whole genome linkage screen. And this was kind of improved genomic coverage compared to that 2006 study. And so what we did is, and I probably should have brought a picture, but we have these chips, and they have these little fluorescent probes on them, and those little fluorescent probes are basically genetic markers across our whole genome. So you can think about them as kind of landmarks, where they're located on different places on different chromosomes. And we use that information, and basically what we're really trying to do at the end of the day is to take the individuals who are affected with Chiari malformation and find regions of the genome that look similar in those individuals compared with individuals who do not have Chiari malformations. But our study actually focused on what we call multiplex families, and that means families that had multiple individuals affected with Chiari. And the reason we did that was, again, we were trying to enrich for the possibility that there was a genetic basis in those families. So our study population consisted of, or the criteria was, there needed to be 2 or more family members who had Chiari malformation with or without syringomyelia. We did exclude known syndromic forms. So for example, I got a question from Dr. Rickay earlier today about EDS, or Ehlers-Danlos syndrome, and we actually excluded those families. So if we knew that there was a formal diagnosis, we excluded those families from this analysis. We collected blood samples to extract the DNA, and we also collected MRI pre-surgical when possible. And so that chip I was just talking about, it had over 600,000 markers all across the genome. And so we typed that on over 350 individuals from nearly 70 families. And then we did what is called a qualitative linkage analysis as kind of a first pass. So what does that mean? What that means is we defined Chiari malformation as simply a binary outcome, if you will. You either have the condition or you don't have it. So there are only 2 possibilities. You're either affected with Chiari or not. And then this question actually came up earlier this morning too, I heard, about modes of inheritance. Well, when we look at the families that we have collected at Duke, a number of our families appear to have what looks like an autosomal dominant inheritance pattern. It's not always a pure autosomal dominant inheritance pattern, but most of the families look similar to that. So that means you only need one copy of the risk allele to exhibit the disorder. So that was the assumption we made. That doesn't necessarily mean it was right, but that was the assumption we made. We also assumed it was a rare condition, and we also allowed for the possibility that the gene which was causing Chiari in one family could be different from the gene causing Chiari in another family. So we did our first analysis, and then we tried not to get depressed because we really didn't see anything that really jumped out at us. So what we wanted to do was to say, how can we improve our analysis? What can we do? Because we know there's probably genes in these families. So we stepped back and we started thinking about what can we do clinically to sort of separate these families? Are there things about some families which are clinically different from some of the other families, and use that information to help split the data set in two? And in fact, that's exactly what we did. So there is a lot of accumulating evidence that hereditary connective tissue disorders like Ehlers-Danlos syndrome are associated with Chiari malformation. But remember, I told you that we actually excluded families that had that formal diagnosis. So we had families who had some of those symptoms but didn't have a formal diagnosis. And I'll show you that on the next slide. But our hypothesis was that the patients who have connective tissue disorders may actually represent etiologically a completely different mechanism, so this craniocervical instability, compared with what we call sort of the classic Chiari 1 malformation, which is based on this cranial constriction hypothesis. So how do we decide who qualified as connective tissue disorder positive? So these are some of the symptom presentations we looked for within our families. And again, these families did not have a formal diagnosis. That doesn't mean maybe they didn't have Ehlers-Danlos, it's just that they had never received a formal diagnosis. So we were very fortunate because when we did a stratification, this split our data set almost exactly in half. So from a statistical perspective, that was quite helpful. And so now all of a sudden we weren't so depressed because now we're starting to see peaks. So that's what we were looking for. So what you can see here, let me see if I can get the pointer to work. What we're looking for, our magic number with this particular analysis is 3. We were looking to obtain what's called a Lodge score of 3, which means the odds that we're observing this association are 1,000 to 1 that it's just due to chance. So those are pretty good odds. So what we saw, particularly in the connective tissue disorder negative families, that's where we were really seeing the best evidence for linkage. And so you can see one peak here on chromosome 8, and there's another peak on chromosome 12. And I'll tell you why we were interested in that. So both of those regions where those peaks were contain genes called GDF6 and GDF3. And those genes are part of a family of genes which are called growth differentiation factors. And they're part of this pathway that is known to regulate bone patterning during development. So that sounded like those were really great candidates to us. So the other thing that was really helpful and sort of gave us a few more clues that we might be on the right track is that these two genes had already been associated with a number of different clinical symptoms and phenotypes and over syndromes like Klippel syndrome. And Klippel in particular was very interesting to us because Klippel involves fusion of the cervical vertebrae and, more importantly, a small proportion of Klippel file patients are actually, or Chiari patients have Klippel file. So these seemed like great candidates. So then, remember, we had just taken snippets, or SNPs literally is what they're called, single nucleotide polymorphisms, across these families, and we had found these regions. So now what we're going to do, those were just landmarks. So now we're going to go in and we're going to sequence those genes and look for changes. And in fact, we were really fortunate. We actually did pick up changes. This is the formal change, A249E. That's a specific mutation that we found in 3 patients from 2 families. And this was important because this particular mutation, we checked 2 different databases, and in one of the databases, we didn't find it in controls at all. And then in another database, it was very rare. And now, remember, we only had looked at 66 families, right? So 2 out of 66 families had a mutation, this mutation in that gene. So GDF6. So that was a very high increase compared to what we'd seen in controls. So we were also very fortunate because some other people had originally found this mutation in other disorders, and so they had done a little bit of functional work to see what does that mutation do to that protein. And what they were able to show is that it actually reduced the amount of that protein which was produced pretty significantly. So that also was comforting. Now, we thought maybe we were on the right track. But I will say that this is a very complicated mutation, and it's a very, very complicated gene, because just because you carry this mutation, it does not mean you're necessarily going to have Chiari malformation. So it's what we call reduced penetrance. And then not only that, but this mutation gives rise to a number of different types of phenotypes. So it gives rise to ocular phenotypes as well as skeletal phenotypes. And we really have no way of predicting at this point which that's going to be. And even within the skeletal phenotypes, I'm going to show you on the next slide, you'll see that, sometimes it manifests as Chiari 1, sometimes as Chiari 0, and not all the affected members in a family necessarily have that mutation. So these are the families that we found the mutations in, and the minus is the mutation. The plus is the usual variant, which is found at that particular position. So we even, just focusing on this panel right here, what you will see is that this affected individual carries the mutation. His mother does not carry the mutation, but she actually had Chiari 0. Her sister had Chiari 1, and she also did not have the mutation. So you see how complicated this is. Based on all the biology, it tells us that this gene is really important. But again, it just kind of further underscores the fact that the genetics of Chiari are very complicated. So thus far, this was the largest genome screen that had been conducted in Chiari, and even this is pretty small by standards to other conditions like diabetes and heart disease. So we need more families and more patients to participate. We did see that, trying to think critically about the clinical presentation and how that might be related to the biology of the disease can really improve our information about which genes might be involved. So, and I put here, this work has been published, so if any of you are interested in reading more, you can check it out. So, I described that was what I called a qualitative linkage analysis, where you either had Chiari malformation or you did not. The next thing we wanted to do was to do what we called a quantitative linkage analysis. And that's where we actually use a phenotype that has more variability. So think of something like height or blood glucose measures or weight. You know, there'll be a range of values for people in the room, right? And so those types of phenotypes actually give us more statistical power. So our next approach was to take that type of a statistical approach, and we wanted to focus on the cranial morphology. So I don't have time to get into this. I'll give you the reference later. You can look if you're more interested about the specific measures and so forth that we took. But to suffice it to say, my graduate students, some undergrads, our neuroradiologists spent a lot of time going over lots and lots of MRIs and taking lots and lots of different measures. So we had a lot of information on our families. And I just want to point out right here that a lot of these measures were highly correlated. So I don't expect you to read anything. The only thing I want you to take away is there's a lot of blue. And that means that a lot of these measures are very highly correlated. So what we wanted to do is take a statistical approach called principal components analysis to essentially take all those individual measures and see if we could create some composite measures that would, perhaps tell us more information. So we did that, and we came up with a number of different principal components, but really there were 6 that basically explained over 85% of the variability in all of those measures that we had taken. So we focused on those 6. And I just showed these to you here, really just to point out that each of the principal components is measuring kind of a different portion of the posterior fossa. So the next thing, so we've got these measures now, and the next thing we want to know is, are they heritable? So that was the next analysis that we did. And this table, I know, is unreadable, but I'm going to point you to the important things. So at the top, what you'll see here, this was the heritability estimate for posterior fossa height, okay, which is shown here in blue. That was the most heritable phenotype that we found in our data set. And it was about 77% heritable. That means 77% of the variability in posterior fossa height is probably attributed to genetic variation. Now, and I don't want the neurosurgeons to yell at me, but at the other end of the spectrum, tonsillar herniation, although it is highly associated with affection status, it was not at all heritable. So as a geneticist, that's not a phenotype that we want to be focusing on. Tonsillar herniation isn't going to help us find the genes. So I'll tell you what we did find. So using posterior fossa height, again, what we ended up finding was this nice peak, and it's over that magic threshold of 3 right here on chromosome 22. And there was another good candidate that fell under that peak, that region of chromosome 22, this EP300 gene. And so we thought that was a very interesting gene because it's involved in this complicated protein complex that's involved in regulating a collagen, which is involved in chondrogenesis. And previously, both EP300 and this CREB-BP gene had been implicated in Rubinstein-Taybi syndrome, and that has also been previously associated with a number of different cranial cervical complications. So And just of interest, kind of connecting back to EP300, when we looked at a similar subset of families with that large posterior fossa height, we did see a little bit of evidence for linkage on chromosome 62 in a region which contained the CREB-BP gene, so one of its partners. And then also located on chromosome 22 was another interesting candidate, this ATF4 gene, which interacts with the EP300 and CREB-BP. So you see, this is really very interesting. These are a lot of proteins which seem to be kind of working together. And this particular gene is involved in osteoblast differentiation and function. These are the cells in the bones. And then there's another gene, ATF3, which is related to ATF4, that was also found in one of our linkage regions. So, you know, we feel like we're starting to get an accumulation of information about certain genes. So, in summary for this section, we did find that most of the cranial morphology in the posterior fossa region is heritable, and we did find a number of different chromosomal regions that look interesting that we decided to pursue, and it just sort of underscores, again, the heterogeneity or the number of different genes that must be involved in this condition. And so this work that I just described is published in this paper if you want more information. Okay, so now we did these two different linkage studies. Do I still have time? A little bit. Okay, I'll try to go quick. Okay, I'll try to go quick. There's not a lot to say from this point forward. Okay, so we've got these two different linkage screens, and now what we're trying to do is see if there's coding mutations or mutations in those candidate genes. So there were a number of different linkage peaks, and they were often very large regions, and we couldn't cover everything. And in fact, we ultimately only was able to sequence less than 0.5% of the regions that we felt might be implicated. But we did what's called a targeted next generation sequencing analysis. And we focus on some of these regions here. We prioritized 31 genes, primarily based on biologic function, that they might be involved with connective tissue or bone formation or other conditions similar to Chiari. Of interest, as you probably heard through some of my descriptions of some of those genes, they all seem to be part of this larger connected pathway. And so we did try to sort of focus on genes which might interact with each other, because we thought that might suggest that those genes truly are involved. So I'm going to cut to the chase because I know we're running out of time, but the bottom line is we ended up not really finding any smoking gun, which is very unfortunate. But, you know, we did only look at, 0.2% of the genomic regions. So, we thought we did a great job prioritizing certain genes, but maybe we didn't look at the right genes, so we need to keep looking. The other thing I want to point out is when we designed that sequencing study, we were looking for mutations in regions of the gene which coded for the proteins. But one of the things that we know and we're learning more about in genetics and genomics is that, it's not just the coding regions of the genes which are important, and in fact, how those genes are regulated and turned on and turned off, that those regions of the chromosomes are actually quite important as well. So it is quite possible that a lot of the genetic basis of Chiari malformations may be regulatory in nature and not the actual protein itself, but maybe how much of the protein is produced. So just to kind of summarize everything, we do think that using these morphometric traits is probably going to be very helpful, at least for genetic analysis. And we still think that despite the fact we didn't find anything yet, that, we were encouraged that there were some very reasonable biologic candidates that popped out of our analyses. And our future efforts are going to be to continue to collect more multiplex families. So if you guys are part of a multiplex family and you would like to participate, please come see me and we'll see if we can get you in the study. I see some volunteers from the back now. We're going to continue to do these more careful cranial morphometric analyses. And one of the big challenges and one of the exciting things our group is really thinking about right now is how we integrate information from the DNA with information from RNA. So a bunch of different types of genetic and genomic information. So we think if we try to sort of integrate all these pieces of data together, that may also help us. Okay, now I'll stop. Sorry, I guess I like to talk, but I just want to acknowledge my team and again, the funding sources. Thanks.